Yeah, welcome to my next lecture on biology for all. That is, the topic is cells. I'm going to talk about cells in this lesson. Okay, this lesson is exclusively designed for all the biology students, board examination, as well as comparative related concepts I will give. Okay, let us see. The first thing is, what are animal cells? We'll discuss about animal cells in this thing. That is, uh, actually, animal cells are nothing but all living things are made of microscopic building blocks called cells, and all organism as complex as human. Example, it is made of trillions of cells working together. Each cell contains smaller structures called organ cells, and each of which has a specific function. Okay, and moving on to this discussion, we have key facts. What are the key facts in this topic? Are there? Are nothing but all living things are made of tiny unit cells. The tiny unit cells are called cells. Small units are called cells. And cells contains smaller structures. Cells contain smaller structures called organelles. These are the 2 points, most important points. Are that most cells in animals are specialized to carry out a particular task. Okay. For example, if you take human body cell example, this many types of cells make up of human body cells. Like other animal cells, they have an outer covering or cell membrane and an internal control center called nucleus. Here we can see this complete detailed three-dimensional picture of it in this. This is the actual human cell picture in this. This location is nothing but ribosomes are tiny organelles that help build protein molecules. These are ribosomes. And this location is the nucleus stores DNA, which contains coded instructions called genes that control the cell. This is, while that of this location is called as mitochondria, release energy from simple sugars to power the cell, and this process is called respiration. Another location, this location is called as cytoplasm, is a jelly-like substance that contains cells organelles. It is the site of most of the cell's chemical reactions. Okay, most of the cell chemical reactions. And coming to this location part, a cell membrane surrounds an animal cell and controls which substances can enter or release it. And on this part, we'll call it as organelles or small structures inside a cell. So these are the main things in this discussion. Now, we have specialized animal cells are there. The next one, specialized animal cells are there. We will see about that. And animal develops from an embryo. Its cells multiply and change into many different types of cells. That is, each with a different structure suited to go to do a specific job. This is sperm cells. Long tail for swimming. This long tail is used for, this is the long tail, which is used for swimming. And sperm cells are produced by male animals, and they use their long tail to swim through body fluids to reach female sex cells called eggs. And the next one is nerve cells. Nerve cells. And these are branches of cells. They are specialized to conduct electrical signals, and they have tiny branches to connect with the other nerve cells. The another part of this one is filament. This is called filament. It's muscle cells, and have filaments that interweave, allowing the cells to contract rapidly and so produce muscle movement. See, this is in this specialized animal cells we saw. Here we saw human body cell. So is the most important, crucial information, fundamental required for everybody to study this about cells. So here, what we observed here is we have started animal cells, and we discussed about the key points. One is all living things are made of tiny units called cells, and cells contain smaller structures called organelles, and most cells in animals are specialized to carry out a particular task. And in that human cell, we observe that many types of cells make up of human body. Like other animals, they have an outer covering under cell membrane and an internal control center called nucleus. So we observed about nucleus here, observed about ribosomes, observed about mitochondria, observed about cytoplasm. See, most important points here, cytoplasm is also important point here. Cytoplasm is a jelly-like substance that contains cells, organelles, and it is the site of most of the cell's chemical reactions. And also observed organelles are small structures inside the cell, and cell membrane surrounds animal cell and controls which substances can enter or leave it. This is of that specialized animal cells we saw, that sperm cells, nerve cells, and muscle cells observed that as an animal develops from embryo, its cells multiply and changes into many different types of cells. Now let us observe about plant cells. Actually, in this we have key points are there. Rigid cell wall made of cellulose surrounds cell membrane, maintains the cell shape. So shape of the cell is due to these things. That means a rigid cell wall made of cellulose surrounds the cell membrane. That is the reason behind that. Another one, water is stored in vacuole and in the center of the cell and helping keep the cell plump or turgid. And another one, chloroplasts are green and organelles that capture light energy and use it to manufacture food. And now these are the key facts. Let us see about plant cells discussion. Like animals, plants are made up of trillions of small units called cells. And plant cells are similar to animal cells, but they have a different structure and contain some organelles not found in animal cells. So most important things here we are learning, right? And in this, let us see the details of leaf cell. Like most plants, leaf cells have a rigid cell wall that surrounds the cell and maintains its shape. And inside the cell, a large fluid-filled structure called vacuole. Okay, now this we are observing most important part of this plant cell. That is here. These are the ribosomes, the sites, protein building, and the mitochondria. Release energy from the simple sugars to power the cell, used for respiration. And chloroplasts are the green organelles that capture light energy to make food. This is for photosynthesis information. That means photosynthesis can be obtained, can be conducted using chloroplasts. And another thing here, this entire place, this is storage unit filled with fluid called vacuole and forms central part of each cell. This is the another thing here, cytoplasm. This is cytoplasm is a jelly-like substance that fills with much of the space inside the cell, and this is where chemical reactions occur. Cytoplasm, chemical reactions occur. And regarding this part, this is the nucleus, is the cell control center, and stores cells genes encoded in molecules of DNA. Whereas this part, this other part is called as a thin cell membrane, which is inside the cell walls, control which substances enter and leave the cell. And this part, outer part, this is, that is a rigid cell wall made of substance called cellulose surrounds the cell membrane and supports the cell. So inside, outside. So finally, what are chloroplasts? Chloroplasts are green organelles that capture light energy and use it to manufacture food. That is called as photosynthesis. The phenomenon is called as photosynthesis. In a specialized plant cells, we'll see what are specialized plant cells. Root hair cells, phloem cells, xylem cells. So these three types are there. We'll see in this. As a plant grows from seed, its cells multiply and become specialized for a range of different functions. And the plant cells shown here are specialized to transport vital substances such as water and sugar around the plant. Okay. In that, root hair cells, if you see, they have a long hair-like extension is also there for this, that absorb water and minerals from the soil. So this is most important part of that one, special plant cell. Another one, phloem cell. Phloem cells that transport sugar and around the plant, and these tube-like cells have porous walls that were joined end to end. Another one, xylem cells. Actually, the one-way flow of water. Two-way flow of sugar and pores in end to end is present in phloem cells, and xylem cells, one-way flow of water here observed, carry water up from plant's roots to its leaves, and the leaves have open ends that join together to form a tube. These are the three main important things. So, phloem cells function is nothing but they transport sugar around the plant. This is the most important point. And function of the xylem cells is to carry water up from the plant's root to its leaves. And root hair cells have long hair extensions and they'll absorb water and minerals from the soil. So root hair cells and xylem cells almost do the same thing. That function is same, but that structure is different. One way of flow of water observed here, long hair-like extension for the root hair cells is helps them to absorb water. And coming to the next one, single-celled organisms. We will see single-celled organisms. Now, in this, we have key facts. Single-celled organisms consist of one cell, and sometimes some types of single-celled organisms prey on the microscopic life forms, and some kinds of single-celled organisms can cause disease in humans. Now, I'll see the bodies of plants and animals are made up of billions of cells, but some organisms consist of just one cell. We call these microscopic life forms unicellular, single-celled organisms. and some kinds of single-celled organisms cause disease in humans. Now, can see this example, that is single-celled organisms. Amoeba is the example for it. Most important picture diagram we are observing, that is amoeba is a single-celled organism and that lives in water and damp places. It moves by making its cell cytoplasm flow into extensions called pseudopods and causing cell to change shape. So you can observe here, this is the location that's called as cytoplasm. Jelly-like substance fills the cell, and these are the extensions of the cell are called pseudopods, and this location will call as excess water stored in bubbles called contractile vacuoles. And this is the green part of this one is nothing but swallowed prey is digested inside the small bubbles of fluid called food vacuoles. And this location is the nucleus which stores the genes encoded in molecules of DNA, and the genes control how cell behaves. This is the beautiful three-dimensional diagram of amoeba. Now we will see the different types of single-celled organisms in addition to this one, amoeba. There are many different types are there, and in each type has adaptations to suit this its particular way of life. One is algae, another one is protoctis, another one is fungi. You see in this algae, this location, yellow color part is called chloroplast to capture energy from the light for the photosynthesis. And this is plant-like organisms that can be single-celled or multicellular. Chlamydomonas is a single-celled algae which contains chloroplasts, which are small green organelles that capture energy from sunlight to make food. And protoctis is the tiny beating hairs used for swimming for them, and protoctis are a diverse group of organisms that mostly feed by hunting other single-celled organisms. And paramecium is protoctis that lives in water and swims by beating hundreds of tiny hairs on its surface. The other one is fungi. This is a cell wall observing. Yeasts are single-celled fungi, and they have a cell wall like plant cells but no chloroplasts. And yeast makes breed rise by making bubbles of carbon dioxide during respiration as it feeds on sugar. So most important part of the cell, any cell you observed, that is nothing but chloroplast. The chloroplast is nothing but where the photosynthesis takes place. Chloroplasm. Next, bacteria. Bacteria are tiny single-celled life forms that belongs to category of organisms known as prokaryotes, and they are very common and live in every kind of habitat, including inside and on the human body also. Here every organism is either prokaryote or eukaryote. In this bacteria cases, there comes under prokaryotes, and animals and plants are eukaryotes, bacteria are prokaryotes. Okay, and live on every kind of habitat, including inside and on the human body, that is called prokaryote. Prokaryotic cells, which are much smaller than eukaryotic cells, have no nuclei and mitochondria or chloroplasts. They are called the prokaryotes. The function of prokaryotes is discussed here. That function of prokaryotes is they live in every kind of habitat. Actually, belong to category of organisms. They are organisms, and protective outer coat called capsule surrounds a cell wall. You observed here for bacteria. Here, this is most important diagram. Excellent picture we are observing here. This is genes are carried by closed loop of DNA floating on the cytoplasm here. And some bacteria have a long wipe-like extension called flagellum, and which rotates it, make them move here. This is this entire location. And this pink part is some prokaryotic cells also contain small loops of DNA called plasmids. And this part, blue color part, is a cell wall and cell membrane within it surrounding the cytoplasm. So this is excellent picture of this bacteria. So inside a bacterium, a typical rod-shaped bacterium has a protective outer capsule and a cell wall and thin cell membrane surrounding cytoplasm. You can observe this. We can also take down the notes from this. Next, prokaryotes and eukaryotes, how they are differentiated. See, all the organisms are either prokaryotes or eukaryotes. And here eukaryotes include organisms such as animals, plants, fungi, and many single-celled organisms. But prokaryotes are tiny and single-celled organisms such as bacteria. While eukaryote cells have nuclei and membrane-bound organelles, prokaryote cells are smaller and have no nuclei or membrane-bound organelles. Instead of a nucleus, prokaryotic cell has a closed loop of DNA that floats on the cytoplasm. Float in the cytoplasm. See, prokaryotic cell here, cytoplasm is there, and strand of DNA here observing with the white colored shape. And when it comes to eukaryotic cell, you have nucleus which contains DNA and organelles are there. But in prokaryotes, it is not there. So this is regarding about bacteria cases. Now we will see about microscopes. Actually, these are the instruments that produce magnified images. Just now we observed, and some things are too small to see with the naked eye. Microscopes are instruments that produce magnified images, allowing us to see things in tiny as single cells. And these are used in study microscopic organisms and living cells. So microscopes are used to study the microscopic organisms and living cells. And microscopes are used to produce an image, and light microscopes use light, but an electron microscopes use electron beams. So light microscopes use light, electron microscopes uses electron beams. Now you can see in the left side we have a light microscope is there. The light microscope uses a series of lenses to magnify a specimen tens or hundreds times, and light shines through a specimen and it must be thin or untransparent. This is the picture of a light microscope. Like this. Here, in this microscope part, this part is an eyepiece typically contains lens made of glass with 10 times magnification. And this location is called as, usually 3 to 4 objective lenses are there with magnification such as 4 times, 4x, 10x, 40x, 100x are there. And this location, this part is turning the coarse focusing knob brings simple sample into focus. And another, this location is the movable stage holds the slide. Whereas this is nothing but a mirror or bulb shines light on the sample. And this one is this location is a glass slide holds the specimen being viewed. While that of electron microscope, you see, this device uses magnetic lenses to focus beam of electrons and create images on computer screen. And the beam of electrons has much shorter wavelength than light allowing much greater magnification. And this makes it possible to view organelles inside the cells in great detail. So microscope, using microscope, you can see, like you can see magnified view of a fresh water microorganisms. Using a microscope, what we can do here, see, we can calculate magnification also. Before using microscope, you need to prepare your specimen by placing it on a slide. You can then use the microscope's controls to change the magnification and focus the image. And how to prepare the view, prepare and view a slide here is, I'll show. That is, for example, you take onion cells. Onion cells makes ideal specimens for microscopes because they grow in very thin films that let light shine through them. So in this, the first picture, dice an onion. Use tweezers to peel off a thin film of tissue from the bottom of one piece. Place this tissue on a microscope slide. The second thing here, using a pipette, a drop of water to the tissue, then add a drop of iodine solution, which will darken the cells and make them easier to see. The another picture is telling that slowly place a cover slip over the specimen without trapping any air bubbles and lower microscope's stages and clip the slide into it. The fourth stage, what we will do, how we will arrange it is selecting the object lens with lowest magnification and use coarse adjustment knob to raise the stage to just below the lens. And the fifth stage is nothing but switch on the light and look through the eyepiece. Now use the coarse adjustment knob to focus the image. Another knob called fine adjustment knob, sometimes used to make small adjustments. Elodia plant here, this is. And now we can observe how this calculation can be done using magnification is. You can calculate the magnification of an image using a formula here given. For example, if a cell of is 40 millimeters wide in a magnified image, but the true cell actually is 0.1 millimeter wide, then magnification is image size by true size. Image size 40 millimeter by 0.1 millimeter, then you'll get 400. So it is called for magnification. By rearranging the formula above, you can calculate the true size of the object also. That is the magnified image plant is, suppose if you take this is 20 millimeters long and the magnification 100 times, then true size is equals to image size by magnification. Image size given is 20 millimeters by magnification, that is 100, gives 0.2 millimeters. So that is the true size of the cell. Next, so this is the first problem. Leaf cell magnified 400 times. Answer. Like this it will be the second problem. Tip magnified 40 times. The cell is a leaf which is packed together to form tissues, become visible under this magnification. Magnifying images, you can see here, you can change how powerfully microscope magnifies, switching between its different objective lens, and figure out total magnification and multiply the power of eyepiece by power of chosen objective lens. Example, if eyepiece is 10 times magnified and the objective lens is 40 times, then total magnification is 400 times. Okay. So regarding the microscope, it's the most important device to understand the sizes of the cells. Elodia plant image okay, observed here. Now we'll see stem cells. The next thing is called stem cells. Stem cells are the cells with potential turn into many other kinds of specialized cells in organism's body. Scientists hope that studying stem cells will lead to new cures for many diseases. That's why we are using this cells for curing diseases. A stem cell is actually unspecified cell. It has a potential to divide and become one of many other types of cells. That embryonic stem cells can form any type of the cell, but adult stem cells have more limited potential. And stem cells reach could lead to new treatments for disease, but using embryonic stem cells is controversial. Yeah, this location of stem cell here, if you observe, the cells an embryo are undifferentiated, which means they have not yet formed specialized body cells. Embryonic stem cells at a few days old, an animal embryo is nothing more than a ball of stem cells. These cells have potential to divide and turn into any of many different kinds of cells in the animal's body. Now we can observe here stem cells research, pros and cons. Stem cells have great potential in medical science. Adult stem cells are already used in bone marrow transplants, which help treat cancers of blood. That is called blood cancer, is called as leukemia. Embryonic stem cells might one day be used to replace faulty cells such as damaged nerve cells in spinal cord or cure conditions such as paralysis. And coming to the another one, tissues grown from patient stem cell is not rejected by immune system, unlike transplants from a donor. Consequences. Some people think it is wrong to use human embryos to research or medicine. In some countries, embryonic stem cell research is balanced, and stem cells grown in laboratory can become infected by viruses, causing disease if they are transplanted into a patient. So many different ideas here we have shown regarding the stem cells. So stem cells are potential to turn into many other kinds of specialized cells in organisms' body. Next, stem cells in animals. How that? See, stem cells is a cell that has not yet become specialized, and it has a potential to multiply to make more stem cells. These new cells are then differentiate or become specialized to serve a particular function, such as carrying oxygen around the body or fighting disease causing bacteria. So they are useful. Adult stem cell, you can see here, this is the location called bone marrow, and this is platelets. These are white blood cells. These are red blood cells. Here, what is the use of this one is, adult stem cells are found in various parts of our adult human body. That is bone marrow, you can see. Intestinal lining, you can see. And they can divide an unlimited number of times, but gives rise to only limited range of cell types such as blood cells. Whereas embryonic stem cells, you see, embryonic stem cells are found in embryos in first few days of development, and these cells can develop into all the different specialized cell types that makes up a human body. That this is embryonic cell. nerve cell, skin cell, red blood cell, muscle cell, liver cell. So in all these locations, these embryonic cells can be observed. Next, coming to stem cells in plants. Actually, plants have clusters of stem cells in part called meristems. These allow this meristem at the root tip, meristem in shoot tip. This is top level. And these allow plants to continue growing and changing shape throughout life, unlike animals. So animals, they don't have like this. So why plants are growing like this is because of this meristems. And meristems are found in shoots, tips, buds, root tips, and around the stem. Plant cuttings that contain meristem tissues can grow into completely new plants. So when you cut it, again new one will come. That's cutting grows new roots from meristem tissue in the stem. You can observe here in this picture. Next, mitosis. Mitosis, here this is the picture. The cells in animals and plants divide into produce new cells for growth or to replace cells that are damaged or worn out. This process of dividing to produce genetically identical cells is called as mitosis. That means dividing cells for growth or to replace cells that are damaged out. So that is called as mitosis. Splitting into two. Suppose during the last stage of mitosis, cytoplasm narrows between the two newly formed nuclei and the cell, then splits to form two daughter cells. Mitosis produces millions of new cells in the human body every second. Rapidly dividing cells are found in your skin and the roots of your hairs and the bone marrow where new blood cells are made. This is the picture in this. Each cell nucleus carries a full set of organisms genes. This location in this picture is cytoplasm narrows between two newly formed nuclei. So above one is there, below one is there. Picture you can see this is a newly formed nucleus. So how the cell cycle works? Mitosis is the final stage of the cell cycle and the process of cell growth and division. Before mitosis begins, cell makes a duplicate of all the DNA stored in chromosomes in the nucleus. So during that mitosis, duplicate chromosomes are pulled apart. After mitosis, each daughter cell grows and cell cycle is repeated. So what is mitosis here? A small picture to understand cytoplasmic division here itself and nuclear division, and cell grows and chromosomes are duplicated here. This is the thing we observed about mitosis. Next, how do cells divide? That means the process of cell growth and division is nothing but cell cycle. Earlier in the key fact of this mitosis is called final stage of the cell cycle. Mitosis produces genetically identical cells and leaves the number of chromosomes unchanged. During mitosis, what happens? Nucleus of the cell divides into two nuclei, which two identical sets of chromosomes. Mitosis has four stages in that case. The one stage is called as prophase, metaphase, anaphase, and telophase. Before mitosis, what will happen? DNA in a cell is stored in structures called chromosomes in the nucleus. Before mitosis, these look like long and tangled threads. These are. And the second thing is prophase. The membrane enclosing the nucleus break up. The DNA molecules wind up tightly and making chromosomes condense, shorten and thicken. The chromosomes look X-shaped because here each one attached to an identical copy. Each side of the X called chromated. So chromated here. Metaphase. Chromosomes are moved to the center of the cell by microscopic fibers, spindle fibers. Spindle fibers. See, this is. Next, anaphase. The spindle fibers separate each chromosome into chromatids and pull a set of chromatids to each end of the cell. Anaphase. Now I'll see telophase. Nuclear membranes form around each set of chromatids and cytoplasm begins to divide to form two separate cells, that is cytokinins. Cytokinesis, that is. Cytoplasm division is cytokinesis. So these are the most important points we have discussed. Observe the two nuclei in the picture also. This we have observed. Now we will see what is meiosis. Sexual reproduction involves a special kind of cell division that is called as meiosis, which takes place only in sex organs. It produces sex cells which have half the usual number of chromosomes but unique combination of genes are there. Unlike mitosis, meiosis is a special kind of cell division, produces gametes, they are called sex cells. After meiosis, cells have half of the usual number of chromosomes, and meiosis gives each gamete unique mix of genes making each offspring different. Crossing over. In normal human cell, half of the chromosomes comes from person's mother and half from the father. During meiosis, these maternal and paternal chromosomes pair up and swap random segments of DNA, produces process called as crossing over. This is a segment of DNA swapped between chromosome pairs here. Now, how this meiosis will work out? Meiosis has two rounds of cell division. Observed. The first, crossing over takes place and the number of chromosomes solved, and second, each chromosome is pulled apart into two chromatids as mitosis. The first thing is, before meiosis begins, arms of each chromosome duplicate and making the chromosomes X-shaped, and each chromosome now consists of two identical copies called chromatids. Next, second thing is now the X-shaped material, maternal and paternal chromosomes pair up. The arms of paired chromosomes cross over and swap segments of DNA. And this third part here is the cell divides one chromosome from each pair goes into each daughter cell, and each chromosome now carries a mixture of genes from two original parents. And the next one is a second cell here. This thing is a second cell division occurs separating each chromosome into chromatids. And the fifth point in the working of meiosis is cells develop into sex cells and sperm and egg cells. Each has half of the usual number of chromosomes. When a sperm joins an egg cell, the usual number will restore. This is regarding the meiosis and mitosis differences. Moving on to the most important point in this cell biology, that is binary fission. Prokaryotic cells such as bacteria divides a process called binary fission, which means splitting into two. Binary means two. Binary fission is the main way that bacteria reproduce, and it allows them to multiply quickly. And that key facts: bacteria use binary fission as a form of asexual reproduction, and a bacterium cellular DNA is copied before the cell divides. Another thing is binary fission produces genetically identical cells. See that identical cells in the diagram, you can see how beautifully we have shown. Dividing bacteria. This electron microscope images here, these are E. coli bacterium dividing. This is, and bacteria have no cell nuclei, usually have just one or two chromosomes. Each chromosome is closed loop of DNA. Each daughter cell receives a copy of the original cell's DNA. This is, a new cell wall forms between the two cells. How binary fission works, we'll see. The circular strand of DNA carrying bacterium's genes duplicated here like this, and after that cytoplasm begins develop to divide. The new cell wall forms the middle here. This is the two daughter cells separated after that. So 3 points here very clear, that first strand is how dividing shown, after that duplication takes place. Now we'll see calculating the number of cells. How? In ideal growing conditions, bacteria may divide as often as once every 20 minutes, allowing them to multiply in number quickly. If you know how long it takes for a new bacterium to grow and divide, you can use the formula used here to calculate how many bacteria a single cell can produce. Actually, the formula you have to use is number of cells is equals to 2 power n, where n is the number of generations. So this is calculating number of cells formula. For example, how many bacteria could a single bacterium produce after 3 hours if division time is 30 minutes? First point here, the calculate how many divisions can fit in 3 hours. So 3 hours means 180 minutes by 30 minutes. That number of generations is nothing but 6 here. And then work out the number of cells produced. The number of cells produced is 2 power 6. 2 power 6 is 64. So 64 is the answer, that is number of cells. Next, culturing bacteria. Bacteria is other microorganisms, and other microorganisms usually can be grown and studied in a lab to prevent contamination of sample by unwanted microorganisms. It is most important to carefully follow the set of procedures known aseptic techniques. In that key fact, we have bacteria grown in culture mediums such as agar jelly, and bacteria should not be grown at temperatures higher than 25 degrees. Aseptic techniques help prevent samples from being contaminated by unwanted microorganisms. Agar plates, how can be studied? You see, bacteria grown in a nutrient medium and a solution containing all the nutrients they need, and this solution is often mixed with the hot agar jelly, which is then poured into plates called petri dishes and allowed to cool and set. Bacteria sample is transferred to the surface of jelly with a in, insulating loop and a loop wire that is streaked across the surface of the agar. Here can observe here how it is there. And not only that one, the petri dish is kept in warm place for several days. The individual bacteria from the sample multiply to form visible spots called colonies, and each of which contains millions of cells. And this part of the diagram, this is the wire inoculating loop is used to transfer a sample of bacteria to the agar plate. And in this, agar is a solid jelly that is mixed with the nutrients that bacteria can feed on. Now we will see aseptic techniques. Aseptic techniques helps to prevent a culture of microorganisms from being contaminated, unwanted microorganisms from the air, dirt, and your body. Inoculating loop here, this is Bunsen burner, tapped petri dish, this is reusable equipment such as inoculating loops are sterilized with the heat and allowed to cool before use. And sterile petri dishes are opened near a Bunsen burner flame. The heat of the flame draws air upward and preventing dust from settling on the plate. Petri dishes are tapped shut to stop microorganisms from getting in. After bacteria transfer to petri dishes, the dishes are kept upside down to keep condensation from dripping into the agar. Samples of bacteria are cultured at 25 degrees Celsius, a temperature too low for most disease-causing microorganisms to grow. These are called aseptic techniques. Now we will see effect of antibiotics and antiseptics. Effect of antibiotics and antiseptics. Most important topic for common for some of the subjects, biology and other chemistry subjects. Antibiotics and antiseptics are actually chemicals that are harmful to bacteria. And this experiment tests the strength in antibiotics by using them to inhibit the growth of bacteria on agar plates. These are agar plates. Here inhibition zone shown is free of bacteria. Another one here, this white part is a paper disc soaked in antibiotic solution. Now we'll see the method of understanding how the antibiotics and antiseptics will affect them. Is first we have to prepare agar plates are covered with evenly with bacteria using either a dropping pipette and spreader or cotton swab. And another second stage is the paper discs that have been soaked in an antibiotic solution are placed on the agar plate. And the third stage is we have to add paper disc soaked in a distilled water rather than antibiotic solution to a second plate, and this plate is the control. Any difference between the two plates will be due to the antibiotic and not on the paper. Now you can see the size, that is incubate the plates at 25 degrees Celsius for 48 hours, then measure inhibition zone around each colony. This is about method of observation of doing this experiment. And what is the result in this one? The result is the antibiotic on the small disc, actually bacteria colonies growing on agar, showing here small disc of paper has seeped into the agar jelly, inhibiting growth of bacteria and so causing clear circles called inhibition zones. And the stronger the antibiotic, larger the inhibition zone. How will you calculate the area of inhibition? Let us see. The inhibition zones on the agar plate are circular, and so you can use the formula for area of circle to calculate the size of this. This is area is equal to pi r square is the formula, where r is the radius. Without opening the disc, use a roller. This is the roller to measure the diameter of several inhibition zones and then figure out the average diameter. Halve your result to find the average radius and put the figure of the average radius into the formula to figure out the answer. Okay, dear students, this is about cells biology. We have discussed today. We have discussed all this information, that is binary fission. What is binary fission, and what is the culturing bacteria? What are the effects of antibiotics? What is meiosis? Meiosis. What is the mitosis? What is meiosis and what is mitosis? And also we have studied about different kinds of stem cells. What are stem cells and animal cells, plant cells? Using of microscope also we have studied to see these cells. And bacteria case. What is exactly bacteria? Whether it is single celled or multi-celled and all we observed. Amoeba discussion as single cell organism. Amoeba is also observed. And are observed about plant cells and different things in the plant cells, where this mitochondria, what is the function, chloroplast, what is the function, ribosomes, what are the function, and very important diagram observed of the plant cells, of that specialized animal cells, and mainly human body cell example, animal cells example is key facts also we observed. This is today's class on the cells biology for all. We have completed. I hope you all understand this lesson, and from this video you can take down the notes also by keeping the video pause, and you can study it for the examinations. Okay, thank you very much. We'll meet in the next session with more beautiful presentations. Thank you.